think this is even more like. What's this one?
A couple of them.
Yes?
Yeah.
Cool. Can everybody also hear me online? Yeah? Cool. Yeah? Okay, then I will start, right? Yeah? Okay. Hello, everyone. I think yes. Yeah. So I'm Kevin. I'm a PhD student of flora, and I've been working here for a little bit over 4 years, and I will defend soon. And today we'll talk a little bit about reconstructing flora from ancient sediment DNA. We cover some basics and some general introduction. Then we will talk a little bit about Monticchio. And we will close everything off with talking about spruce in Scandinavia. So we have a lot of artwork in the presentation, and all the artwork has been done by Jill Pelto, so you can see the link underneath this slides. Cool. So we just go straight into it. So I already talked about this already. So we talked about what is sedimentary ancient DNA. 13,000 years of flora change in Monticchio, and the post-glacial recolonization of spruce in Scandinavia. So to start off, what is ancient sedimentary DNA? You see a very nice sediment core here in the bottom. And if you zoom this in a little bit further, you can see a lot of things happening here. They look very nice. There's a lot of layering. So every layer more or less corresponds to a year in this case, I think. So there's annual layering, and there's also different coloring from different sediment types. And the one that we highlight here in this color are layers of diatoms, so algae. So why are we looking at sediments? Because we are interested in the past, and sediments are annually deposited. So if you imagine this lake, you have sediments in the bottom, and over time, things coming from the catchment actually end up in the lake, and they deposit their material on top of the previous layer, and so you get the annual increase in sediment. So if you progress this to time, things end up in the sediment core. So if you progress this to time, every year or every certain amount of time, there's an increase in sediments, and this is giving you a temporal sample that you can use and date and use to see what is around the catchment of the lake. There's two components here, that there are things that are in the catchment, so the grasses and the small flowers, and there's also trees in the background, which is more a regional signal.
They can't see the slides.
They can't see the slides?
No.
So what do they see?
It's the first slide.
They're still at the first slide. Okay. This is sharing my screen, right?
Presentation could be
Restart the presentation.
Or maybe it's like
No, you.
So what are you seeing now?
the same thing as you.
Same thing as me?
Maybe it's because you're sharing the. Let's see if that makes sense. No,
Now you see nothing? Or
it's the same thing as before. So it's like the PowerPoint before the show. Now it's working. Yeah.
Now it's working? Cool. We tested this in the
Try
afternoon.
to change to go back.
No, keep it like this.
Now it's
Slides.
Oh, but now I can't change the slides. Oh,
Okay.
now it goes.
Now it's good.
Now it's working? Cool. So just go to the beginning then, like here. I think this is a good, really like the transition thing.
You want me to?
No, I think it's okay. I'll just hide myself for the viewers. Yes. Okay, so we'll go back.
Why sediments?
Why sediments? Exactly. So sediments are in the bottom, and annually they increase and they reconstructing. So they're in the catchment, right? So if you progress this, which you can now very good see online too. Yes. There are two different components here. Like you have the regional component of what is ending up in the sediment. So they have these trees in the background that produce massive amount of pollen, which can transport a long distance, and they can end up still in the lake. While the things that are in the catchment, they produce, they also end up in the lake. So it's a combination of the two, basically. And through time, compositions of flora changes, what's happening in the catchment and regional things. And this ends up as a sediment core that looks like this, and we can go back in time and see what is present in the past. So that is a real sample. This is an interpretation in artistic way, which shows very nice the layers, and the further you go down in time in the gray zones, that's less organic because there was less plants and things in the catchment, and the more you go to the top, the more recent it becomes, and the more things start to appear. So if you kind of zoom in into this, you can see there's a lot of different pollen, different seeds, and different macrofossil remains. And you can look at this under the microscope and reconstruct what is present based on what you see, which is kind of the traditional way that this has been studied for a very long time. But now we are in the DNA period, so we are not really looking into the microscope that much. We are taking the sample and we extract the DNA from these sediment samples, because DNA is also present. It's bound to the mineral particles. You can't really see it, but we know it's there. We can get it from the samples, and we can use this DNA to actually also reconstruct what has happened in the catchment on top of the macrofossils and the pollen. So how this looked like in real life, we get the sediment core, which is one of the cores that we got from Monticchio. This over here is actually Laura subsampling the core in Potsdam, I think, in Germany. So basically, we go into the core, we prepare, or we are making a clean setting so we don't contaminate our own samples, and then we slide different amount of material into this tubes, and that we put straight into the freezer because we want to kind of keep it cold so that the DNA doesn't degrade very quickly. And these tubes we bring into the DNA lab or into the laboratory. They look just basically like tubes with dirt when I get them, so they don't really look like cores anymore. They look all the same to me. Yeah, and then we are in the laboratory and we wear all these protective suits, and we glove everything, we clean everything with bleach so that we don't get stuff from outside into our samples. And the question, of course, is like, what is ancient DNA and why do we need to be so careful? Like, why do we need to wear like almost spacesuits to do our work, right? So if you picture DNA, we usually picture this double-stranded, double helix kind of thing, right? But if you kind of make it 2D, then it's 2 strands of DNA, and this is like high quality. This is like not degraded or nothing has happened to it. But as soon as it's deposited in the environment, I will slow down talking a little bit. So as soon as you kind of get it in the environment, your DNA doesn't really stay preserved, or it doesn't really stay like this. So over time, your DNA starts to fragment. It becomes smaller and smaller. And in the end, if this takes a long time, then you end up with very short fragmented DNA that is both fragmented, and they also have some chemical changes which causes them to have some damage in the fragments. So ancient DNA is highly fragmented. And the question that kind of pops into now, like how far can you go back with this, right? Because if it degrades so fast and becomes smaller and smaller, like what is the time range that we can use with this? And very recently, and so this is the beginning of the year, they pushed the boundary beyond 1 million years. So they used mammoth bones that were dated to be a little bit over 1 million, and they were able to get DNA from this and be able to authenticate this. That is definitely 1 million years old. This is really pushing the boundary, I think. I'm not really sure how much further we can go than past this.
This was teeth. Okay.
Teeth.
So it's even
Yeah.
better.
Yeah. Under the best circumstances, it can be up to 2 million. I don't think this has ever been really done in sediments, although there are some records that are going 1 million years where they did find some stuff, but it's not really confirmed yet. Yeah, so do need to talk a little bit about methods. We'll just have one slide. So what we do is something called metabarcoding, where we look at one specific gene for plants, which is in the chloroplast. And every plant, basically that has a chloroplast has this gene conserved in it. And what we do is we have one particular gene, and then the gene is the same within the taxa, within the taxon, and it's different between taxa. So it's representative with different bars, they have different sizes and different colors. And if we compare this to a reference database of known sequences that we know the origin of, we can identify these DNA pieces to specific taxons. So in this case, like the top one goes to an oak or like a tree, a conifer. And in this way, we can find the DNA. The DNA is super fragmented. We have small bits. We know what we're looking for, so we can identify where it came from. So just to summarize this in a few sentences, is that sedimentary ancient DNA is environmental DNA extracted from annual deposits, including lake sediments, peat records, and cave sediments. Ancient DNA tends to be more local, so it comes from the catchments rather than coming from a very long distance. So it's much more local than pollen. And in some cases, not all cases, DNA can increase taxonomic resolution compared to pollen. So pollen goes to family level, or and then DNA can potentially go to lower taxonomic levels. Yes. Are there any questions at this point? You're welcome to ask any questions. Yes.
Why is it that it's a more local signal?
Because most of the DNA we think, we're not 100% sure, but it's coming from leaf material that's actually dropping into the lakes and things that leaks out from the catchment. So it's much more likely that things from close by are actually ending up in the lake. And there's been a study done in a lot of lakes, top sediments in northern Norway, where they found that most of the DNA signal is actually coming within 5 or 10 meters from the lake, and the further you go away, the less likely actually to be detected.
Essentially, it doesn't come from pollen. The DNA signal doesn't come from pollen.
Well, would that, I mean, I'm just wondering, I mean, isn't it possible that the pollen DNA degrades slower than the leaf DNA? So even though you have more leaf DNA,
I would actually say it might actually go faster in pollen than it leaves. Like pollen, they have all these spores, so the material goes very quickly into the pollen, and pollen has only two cells, right? And one leaf has massive numbers of cells, so there's much more DNA there. Although maybe it degrades faster, you're much more likely to detect it than coming from the pollen. We have tried extracting DNA from single pollen grains, and it is a nightmare. It's super difficult. So we did a lot of tests, and it's very difficult. You can find sediments that have loads of pine pollen, but you don't find any pine DNA. So we are quite sure that in most cases, pollen don't really contribute to the DNA. Any other questions? No, so continue. So we talk about, yeah.
I have a question.
Yeah.
So is there a difference between species, for example, trees? So probably trees not slower than
Yeah, so it's a very good question. We actually don't know this.
But it's not the species itself, but maybe the species release more like say material that's more degradable than others. Yes. So let's say a species maybe has a lot of fruits that are preserved well in the sediment, and another species doesn't. fruits, then you would have a bias that there are such bias behind. Some species are much more present than others, even if the abundance around it is the same.
Yeah, so there might be a lot of variation in this thing. So that's why what we find is not very quantitative. It's not really the same as pollen where you can like go back and reconstruct vegetation. We kind of more can do like flora reconstruction. We can say who's present and who's. I don't think we even can say who's not present, because if you don't find it, doesn't mean it's not there, right? So we can determine presence basically. Yeah. Any other questions that popped up? No. Cool. Now we talk about Italy, Monticchio, where we talk about the last 13,000 years. So for you that don't know where Monticchio is, it's a crater lake in southern Italy over here. And there are two lakes basically. There's the big lake and the small lake, and our sediment samples are coming from the big lake. This is a Mar lake, so it's a lake that was created when the volcano actually erupted, and they are basically the craters that remained of the volcano. And it's a very unique record because it's a continuous record that goes back more than 100,000 years. And there's a massive amount of sediment underneath this lake. It's up to 100 meters, even more. Yeah, so it's a very cool place, and it also has never been glaciated. So there's never been a glacier on top of this. That's why we can go back so far continuously. And these kind of lakes and records are super rare. So it's a very nice place. And we've been there on the SVS excursion. That was in 2018. So that's me and Francisco talking about some oaks around the lake. And some of you were actually there. So maybe remember when we were here. We were talking near the place where we stayed, I think, about the lake. So it has been caught a lot of times since the 90s. So at different places in the lake, different lengths. And yeah, so you can see that the lake itself is actually pretty deep. The deepest point is almost 30 meters, so even more than 30 meters from the water surface. And the amount of sediments is underneath is quite a long time. We don't have the whole 100 meters. Unfortunately, we only have 14 meters somewhere on the top. And these 14 meters are, yeah, that's what we have. And just to remember that we actually talked about this before, because the exact same picture is also on the table over here. It hasn't changed. So we have 14 kind of meter long core, which is all cut in these 1 meter pieces. And the first core I showed in the beginning is actually one of these 14 meters. And we took, I think, more than 160 or 170 different samples along these 14 meters, covering a very long time period. And it has recently been dated that the oldest sediments that we have are actually 32,000 years. So this is before the late glacial maximum. So we'll show some preliminary results. We're still working on this, but we will show some interesting things anyway. So here is kind of the DNA presented. You have the eighth and the. I can actually think you can remove this, right? Put this in the bottom. Yeah, so there's the pictograms are there again. So this is based on DNA bedrock coding. It looks very similar to a pollen diagram, but it's not exactly the same. So all these white bars are the places where we find DNA of these particular plant species. And we can overlay this with, why is nothing happening? Okay, yes. So if you overlay this with pollen, you can see that in most cases we actually record the same taxa.
You tell me when you need.
That's better. Yeah, it's okay by me too. Yeah, so this is just showing the trees. So we have a lot more other taxa, but it's just showing the trees. And because it's a little bit difficult to see what's going on here, so we highlighted some of the cool taxa. So we have oak over here. So we can kind of see that the red line is the pollen. and the white bars and the areas are the stuff that we find without DNA. So what you will see is kind of that in most cases we find the same things. We find oak where pollen of oak was also recorded. There are a few possible species of oak that we can't really distinguish very well. So same with pollen, we can say it's oak, we can't really say exactly which species it is, which can be multiple species in multiple times. And we were actually talking about this with Francisco when we were at the lake. He's holding oak leaves there. Then we have here Abies alba, which is not very abundant with both pollen and DNA, but you can see that the DNA and the pollen actually overlap quite good. So you get kind of the same pattern here that it comes up the same amount at the same time as pollen appears, and the signal drops when the pollen starts to disappear. But then there are some examples where actually the DNA shows slightly different results than the pollen does. So this is an example for taxus. And we actually record DNA from taxus way before the actual pollen starts to appear. Same as when the pollen starts to more or less disappear, we still find taxus in some of the samples. So we kind of find DNA earlier than the pollen does, and we also see that DNA is present a little bit after the pollen disappear. Then there are some cases that we should also highlight is that for these two species, which is Ilex aquifolium and Viburnum, we don't really know which species it is. It can be a lot of different species. They all share the same sequence, basically. But in this case, pollen show very little. It's only a few samples where you actually can see some pollen grains. But in most cases, you don't really see them. But if you look at DNA, it's basically continuous covers at the lake when we look at DNA. And then, of course, there are also some examples where we find whether pollen was found. So Betula is one of these examples. We find a little bit of Betula pollen in the late glacial places, but the DNA is from Betula is actually absent. So in this case, DNA doesn't recover the species, although there is pollen present. Then there are some species or taxa that we can't really distinguish based on the marker that we use. So in this case, Fraxinus and Olea, they have exactly the same sequence. So when we see it, we can't really tell whether it's coming from the olive tree or whether it's coming from Fraxinus. But Fraxinus is a common species that appears throughout the whole core, but it actually disappears in the last samples, which is basically the same as the pollen suggests. So this is all based on one marker on the chloroplast. But we actually also did some kind of the same similar approach, but we use a different marker, which is a lot more species resolute. But it has a problem that it doesn't really amplify a lot of species. So this is a thing that's specifically for grasses. But we had some luck that we actually had some bycatch of trees. So here you can actually see that what we find here is actually Fraxinus and not olive tree. So in this case, we know that at least in this period of time, 6,000 to more or less 1,500 years ago, we know that part of the signal is coming from Fraxinus in the previous case. So maybe you can actually switch the light back on now, or people fall asleep. Yeah, so we detected most of the three species except Betula. There might be some methodological reasons for this, because Alnus was very present. Maybe we didn't really see it very well, but Alnus and Betula are in the same family and they have a very similar sequence. So maybe this part of the Betula signal is actually clustered together with the Alnus. That might be one of the explanations. Another explanation is that actually Betula was not growing near the lake or in the catchment, but actually was growing further away. So the pollen represents a regional signal, while the DNA actually shows the more local signal. In overall, so far in just the Holocene samples, we detected more than 65 different taxa, and a lot of them are herbaceous and aquatic plants, which are usually not very well recovered with pollen because they don't really produce much pollen. But we find them with DNA. So the DNA is very nicely complementing the pollen results. So they don't really DNA doesn't really replaces pollen, but it just shows a little bit of extra information that we can get out of the same samples. So it gives a more local signal. And there are some methodological challenges because I told you that the core was 32 meters that we had, right? So, and we only presented the last 12,000. It's mainly because all the samples are very problematic and there's low DNA concentration and we had a lot of low quality samples there, so I didn't really show it. But we have still a lot of samples that are not really included in this. Yes. So that was Monticchio. Any questions about this? Yeah.
Did you find any species instead of using DNA HMD compared to the one outbred by the
Yeah, there are some taxa, but I don't know exactly which one. So there are some species, a lot of species that you find with pollen and microfossils that you don't really recover with DNA. But we also find a lot of additional species that you don't find with pollen and stuff. Yeah, and also there's a lot of different taxonomic levels here. So some I think is Rosaceae family. We can actually split it in multiple taxa because we have a better resolution here. While the pollen, so it's just the whole family is present. So it's a little bit difficult to compare how many taxa are present in different things because they're all on different levels.
Lots of aquatic plants are found with DNA, not with pollen, because they're not producing. So aquatic plants records are very important. Good signal. Might be surprised if I can not expecting more.
So there is one that I can actually go back. That has been a part of discussion for a very long time. I think there's this. This is the no. So there's Cedrus. So Cedrus is not supposed to be in Italy. Cedrus is a species that occurs in the Atlas Mountains and in Turkey and was present in Europe and Italy in the previous interglacial, so a long time ago, but actually disappeared. But we still find some low abundant amount of DNA. I removed most of the stuff because I have very strict filtering here. But we do find sometimes weird things. And one of the ones that is also a little bit weird is that we find cotton. So this is cotton, the genes of cotton, and a few samples, it's very abundant. And cotton is not supposed to be in Italy 6,000 years ago. So this might actually be a case of contamination that happened from maybe some fibers or something in the sample, and then it kind of overtakes the signal, because as soon as you have cotton in your sample, like most of the trees start to disappear, which is a little bit odd, right? So it looks like these samples are actually, yeah, we're looking into this, whether this is like a bioinformatic problem or if it's really something like this. So we do find a lot of surprises. Yes. Any more questions? Yes.
Yeah, well, I'm just, when I was looking, just, not looking at any of the labels, to me, it looks like a lot of the things actually have a similar
Yes.
shape. And I just, is that what you expected, or is it, I would think that would be kind of surprising.
Yeah. So it is a little bit, this is still a little bit biased towards methodology. So we do very strict filtering. So in a lot of samples, we actually have the signal, but we remove it because we have it, it's above the threshold. So then it's sample quality that kind of determines whether a sample goes down and up and things like this. So in most cases, like it's not very quantitative. So if a peak goes down and up, it doesn't really mean there's more or less of it. It's just that we are more or less confident in whether the signal is really true or not. Kind of. So I don't really in general like the way of presenting it like this because it looks very.
So it's not abundance.
No, it's not abundance. It's more like presence. But there is a lot of signals that are occurring in multiple samples with multiple taxa, which is basically more like a methodological problem than actual biological one. That's my feeling. So we do redoing some of the analysis and building a little bit more confidence in this. Yes.
About the age of the sediment, keep you measuring or?
No. So we have a very long list of collaborators. So we didn't really core ourselves. We have a lot of collaborators working on the same course, and there's people working on the chronology. So because this is actually a very cool lake because it's vast, so you have this annual layering. They have actually counted physically under the microscope the layers for every year, and they anchored it on one of these volcanic eruptions. So you can count of the they're called tephras, so they're like volcanic ashes, and then you can kind of relatively well date this course. Yeah.
No, because my next question would have been like, okay, what about the sedimentary suspension? Because some cases you have a lot of mixing within the sediment that can also
Yes, I don't think there's that much mixing
Yeah,
in
it's
this
okay, okay.
lake. Like it looks very cool.
Yeah, okay,
Yeah.
it's like there's not much.
You can count
Yeah, okay.
32,000 years of
Yes, okay.
layers, one by one. It's the most accurate way of dating. Actually.
Yeah, so they did radiocarbon dating on this too, but actually the lake is producing a lot of carbon dioxide, which is incorporated into the plants, which is actually older carbon than is actually around the lake. So if you radiocarbon date this, the dates are ending up much more older than they actually are. So the radiocarbon dating doesn't really work really well in this case. Yes, but the dating is pretty accurate because it's counted basically. Yeah.
You know, because the working place and
Yeah.
now really mixing the sediments, so I'm just curious about these lakes. That maybe it's really stable.
Any other questions? So we take a break, or they want me to continue?
We go to Scandinavia.
We move to Scandinavia. Cool. Yeah. So the next part that we're going to talk about is another project that we've been working on for a very long time, and Laura has also been working on this specific questions for quite a long time now. And in general, this kind of questions about the spruce recolonization has been going on for a very long time. So yeah, this is one of the artworks that Jill Pelto produced, and this kind of shows. So you can see the line. It's actually real data, and I'm pretty sure it is tree line movements that is incorporated this. I'm not really sure exactly anymore how long the period, but at least it shows some real data. And if you go to the links, you can actually see the description of what the art represents. But we are actually interested in these trees. So these are trees that are actually growing above the tree line, above the current tree line. And this is how they look like in art, but this is how they look like in real life. So they're kind of trees that are not very tall. They are very relatively small. They grow above the tree line, so you can basically see that there's not that much going on around it. This one is even more spectacular. So they usually, the spruce trees usually, if you go to the forest, they are massive, like 30 meters or 20 meters. They grow in a nice tree, but these are not really trees. They have a trunk, but a lot of their branches are actually laying on the ground. And because of this snow cover, they actually can shoot roots again, and they can sustain themselves for a very long time in this kind of growth form. Yeah, so if you have the tree line or the timber line, then there are some trees in the tree line, and the clonal spruce trees that showed in the picture before, they're actually above the tree line. So we'll introduce you a little bit about the glacial of the post-glacial recolonization of spruce in Scandinavia based on pollen. So present-day spruce is pretty much all over the place in Fennoscandia and Scandinavia. It's also present in the Alps and in Central Europe. But those trees are actually not really, they are related to, of course, to the trees that are in Scandinavia, but they are genetically different. So we already know that these trees didn't really contribute to the recolonization of Scandinavia. So that's why there's the red line. We don't really talk about these trees that much because we know they didn't really contribute. So if you go back to the last glacial maximum or a little bit after, the ice sheet was massive across Fennoscandia, and there was probably not many places where trees actually could be. So the idea was always that spruce was confined to the Russian plains, so quite far into Russia, and that they more or less survived there during the harsher times. And then when the ice retreated, they slowly moved towards Fennoscandia, and over time they made it into Fennoscandia. So this is almost 11,000 years old. Of it, 11,000, where the pollen, if you look at pollen records, there's a lot of pollen increase in this kind of regions in the Baltic states, and it's still Russia. And then, so the beginning of the Holocene, spruce is kind of at the border of Finland, but still didn't really come much further than that. And then after that, it takes a very long time before spruce actually colonized the rest of Fennoscandia. And the time that it reached southern Sweden and central Sweden is approximately 2,000 years ago, based on pollen. Yeah, so it suggests that it's a very late arriving species to Scandinavia. But this idea has already been challenged for a few decades now. So Kuhlmann has been finding a lot of fossils, megafossils, not only from spruce, but also from pine and even some larix in the Scandinavian mountains. And these are like fossils that can even be tree trunks or like fossils that are radiocarbon dated to be late glacial and early Holocene. So basically the oldest fossils that have been found are like 11,000 or 12,000 years old, which is very close to when the ice was still there. There's already macros fossil remains in the Scandinavian mountains, and this has always been quite controversial to suggest that the spruce was already present at this time in Fennoscandia, because the pollen showed that the spruces are very far away, much farther away than where the fossils are. But if you even go further back in time, so Tage and Nielsen in 1935, he already published papers. It was actually in German, written in German, where they found spruce pollen during the late glacial, so around 14,000 years in southern Sweden. And they suggested that spruce was very early present in Scandinavia. But this has after that has been often credited to be like long distance pollen transfer from other places. So although you see pollen in these late glacial sites, it's difficult to say whether they are regional or not, because they can transport for quite a long time. Then there was Lindquist, who also looked into this colonization events, and based on morphology, he would suggest that there might have been also spruce colonizing very early, based on morphology, and he even suggested that there might have been some spruce on the west coast. And this has also been collaborated by finding these early finds in central Sweden, that the closest place where you can expect spruce would be probably on the west side of the ice sheet instead of on the east, because the distance is much more closer. So this has always been a debate, and it's likely that this debate has been going on for almost 100 years now. So we are still in the same debate, and we have been looking at this from different angles than just looking at microfossils and pollen. We actually went into the DNA angle, right? So our question basically was, where is spruce present during the late glacial and in Holocene? And we use, of course, our ancient DNA approach. So what we did is, there is this way of detecting spruce. This is a mitochondrial marker, so it's different from the things I've been talking before, which is chloroplast, and which is aimed to be as much diverse as possible. Here we specifically go in and look and see if we can find spruce. So we don't really look for everything, but we look specifically for spruce. And this marker has basically two options. There are only two variants. There's one small one, which is only present in spruce, and then there is the red one, which is present in spruce, but it is also still present in pine. So that's why there's a pine symbol over there. So if it's blue, we know for sure it's spruce. If it's red, we know it's spruce or pine. So we can't really distinguish between the two. So what we did is we got a lot of materials from different places. So in total, we have 14 different lakes. 6 of them are in Scandinavia. Some of them are in southern Sweden during the late glacial transition. And we have some in Finland and in Russia. So there's a lot of things here in this picture. But basically our samples are coming from Jämtland, where the clonal spruce trees are today. We have some samples in southern Sweden, which are covering not the Holocene, but actually the transition between the late glacial and the Holocene. So they're like 15 to 11,000 years old. Then we have some sites in Finland and also in Russia, and one very far away in northeast Russia. So every column that you see in the figure is basically one lake. And on the y-axis, there's the age, and there's an age gap between. So this is 16,000, but this block actually covers 42,000 years old. So it's actually the previous, before the last glacial maximum. So if you look at the left, where you see Jämtland and central Sweden, we more or less find the spruce throughout the Holocene. So you find it very early in these samples. You find it around just before 10,000. Then we see it continuously, more or less through the sediment core. In all the samples in this place, you actually find the other variant. So we're not really sure whether there's a signal coming from pine or from spruce, and it can actually be both. Then if you go to the sites in southern Sweden, which is actually very interesting because you see a lot of spruce appearing in our samples there. And it's not just one lake, it's actually 2 lakes and different samples, and we see it in consecutive layers. So the signal, although it is very low, we are very confident in that this is actually a true signal. Even more interesting is also that we find spruce in Finland, southern Finland, before the actual pollen start to appear. So this is still 6,000 and almost 10,000 years ago, where we find spruce also in Finland. In southern, in Finnish Lapland, so northern Finland, we have some sediments that are actually paleo shoals. I think they are dated to 42,000 years age, so they are very old. They are before the last glacial maximum. So this is spruce that was present from before the ice was actually there. So they are representing the previous generation of spruce. Basically, we also find spruce in these samples. But if you go further to the right, and we are now in Russia, we have a sample in southern Russia where we actually don't find anything. So no spruce and no pine. And then there's one not even further where we don't find the blue variant, but we only see the red, which is pine and spruce. So basically, to take home from this, we see a lot of spruce in Scandinavia, in Jämtland, throughout the Holocene, much earlier than when the continuous pollen start to account. A lot of these samples, they actually have pollen of spruce, but they're so low abundant that they've always been thought to be long distant instead of local. Well, now with DNA, we can actually say that this signal is probably local, because DNA, seen before, is more of a local idea. Also, very interesting is that we can see it in southern Sweden, which has been debated for a very long time, since Dag and Nielsen that's found the pollen. It's always been said that this was long distance distribution of pollen, but based on DNA, we would actually suggest that there was spruce present in southern Sweden. So the next part of this is that we actually went out and sampled these clonal trees, right? Because they are very interesting to our story, and we wanted to look at the genetics of these trees. So we went on expedition twice with Laura, with SBS money, actually, yes. So it was kind of an SBS excursion, but just for us. So we went out there and we went and sampled a lot of these clonal trees. So this was a picture taken after we walked almost 20 kilometers to this place, because we were had a roadblock in Norway when we couldn't drive any further, so we had to walk for a very long distance. And we didn't really time our sampling very well, because the first time we were at we still had the snow from the previous year, and the second time we went, we were in the first weekend when the snow started to appear, so we were always walking in the snow and rain, and it was very cold, but we have very nice pictures, and it was a very nice. Yes. So probably the most famous of all of these trees is Oltico. I guess most people have heard of Oltico. The date is like 9,550 years, and this is dated based on a fossil that is found underneath the tree. And the idea has always been that these fossils that are underneath the tree, actually coming from the tree that's still on top of it. So this would be the same individual that was there 9,000 years ago. The individual that's still there would still be the same. This has not been proven very consistently, because you're never 100% sure that the fossil that's under the tree is actually coming from the tree. There's no genetic continuity studies done on this yet. So this is basically a fossil means that there was spruce. It's likely the same individual, but we're not 100% sure, right? But still, they're very interesting trees to look at. So in general, Oltico is quite small, and most trees are kind of this size, even smaller. But some cases you can actually see that they can reach some meters. And because now of the climate change and things become a little bit hotter, these trees actually doing pretty well, and they start to expand, and the trunks become higher and higher. So probably at some point, if it becomes hotter, this kind of growth form will stop being, and they will become trees, basically. Yes. So for the people that are not from Sweden, the samples are coming from this place, with Jämtland and Dalarna and Härjedalen in the middle. So there are Oltico is in Fyllefjellet, and in most of these places, populations, there are trees that have been dated and they know they are relatively old, or at least the fossils there that they find are old. So what we did is we went out and we sampled the clonal trees, but not only the clonal trees, but we also sampled the trees that are in the forest, so we can have a straight comparison between the two and see whether they are genetically same or similar, or if they're different. And we did this at all these populations that we showed in the previous slide. So we took almost 140 trees that are above the tree line and almost 130 trees that were below the tree line. And after that, we extracted DNA from this and looked at the genetics. And the idea was to see how the clonal trees in the forest are related, how the clonal trees above the tree line are related to the trees in the tree line. So this is based on modern population genetics. So this is not based on sediments. This is individual trees that we're talking about here and population of modern individuals. So there was no ancient DNA involved in this part of the study. So if you take this take these trees and you look at their genomes and you characterize different SNPs, they are like different positions in the DNA genome, and you can compare all of these individuals basically. So every point in the PCA plot is a different individual. The further they are apart, the more different they are, and if they are very close together, they are more similar. And you can see that, so all these colors, the purple one is Jämtland, the blue one is Härjedalen, and the black one's Dalarna. You can see that there's a very good regional trend here. Like the samples that are coming from the same place, more or less, they cluster together, and the samples kind of show latitudinal gradient, right? And this is an isolation by distance plots, which is basically making pairwise comparisons between all the individuals. So every point is a comparison, and it compares the geographical distance to the genetic distance. So the further the points are away, the further the distance in meters, the more dissimilar they become. So distance is a very good predictor here. And it's very well nicely correlated. So the further trees are away, the more different they are. So this is one way of looking at these different trees. There's another way, which is called admixture, which is basically looking at, you have all this genomic material, and you kind of see how many components are this coming from, and their ancestral components. So if you look at this and you look at the ancestry, then every bar represents basically an individual, and the color depends on which ancestor they come from. So the blue component is very abundant in Jämtland, and the yellow one becomes more abundant when you move towards Salland in further down in Dalarna. So there's also here a very nice trend, and it looks like there are two ancestral components that this places are coming from, of spruce trees are coming from trees that are more grouped in the north and trees that are more grouped in the south. But at this point, you will probably also wonder, like, why don't we see any clonal things in here? Because everything looks more like very gradient, right? With latitude, but not with altitude, basically. So this is full of Fjellet, where all TQ actually grows. These are all the clonal trees that we collected, and these are all the forest trees that we collected. And you can see that this almost copy paste, right? It looks very similar in genetic composition. So what we can say from this is that the clonal trees above the tree line are actually identical to the ones in the spruce forest. Basically meaning that these trees that are above the tree line are not so special compared to just the ones in the forest. And that you don't really need to explain one event happening where all these trees appeared at the same place, because then they would be looking more similar to each other, right? And the idea that trees came from the west in a separate refuge that would be very different from the ones that are coming in from the east, can also kind of refute, because they all look more or less the same, and there's not any pattern between the clonal and the spruce forest, which is a pretty cool result, and this is not what we generally expected to find. So maybe a little bit more interesting than talking about the genetics part of it is, what does it mean, right? So combining the ancient DNA with the modern genetics. So these are a series of maps that kind of represent different slices in time. And this kind of the proposed and suggested colonization events that we think happened based on our data. So every tree, they have different colors, and these colors are representing the ancestral component based on the nuclear data. So we have there's two different groups, and what we see in Dalarna and Jämtland is that we are on the transition between these two groups. So we are at the contact zone between the groups in the north and the groups in the south. Then the black trees, they are not really part of this two ancestral cluster. They are a different one. They are coming from the south, so they didn't really affect the results. And then there are the green ones. It's actually the trees that are now in the Baltics. These are genetically different from the ones in Scandinavia. This is not based on our results, this different clustering, but this is based on a lot of stuff that has been done by Pascal and Martin and other people that have been looking at spruce. So this plot kind of represents our idea of where trees were during the Eemian, which is basically the previous interglacials. This is before the last glacial actually happened, before the glaciers started to appear. And spruce was probably a little bit more distributed than it is today. And this is kind of the idea that these two clusters were already there before the last glacial maximum. Because maybe it's good to tell is that the difference between these clusters don't really look very much, but the amount of time it takes to actually get these two different clusters is very much. So this is not a signal that is caused by glacial cycles, but this is things that happen much farther in the past. So this colonization event is not creating these clusters. These clusters were already there before. Just to get an idea, the trees in Scandinavia and the trees in the Alps, they have been separated for more than 15 or 16 million years. So this is way before the Quaternary actually started and all these ice cycles actually started to appear. And the difference between these two blue and the yellow clusters is probably also predates the Quaternary. So these are things that happened very far into the past. Yes. So if you then picture the things that happened at the last glacial maximum, where you have this massive ice sheet, right? Most of the trees that were there would have been disappearing because there's ice, right? So they are not there anymore. But at the edges, trees might actually have survived. So the idea from pollen is that the trees are actually present more to the east in Russia. But based on our data, we would actually suggest that spruce was actually much more closer to the ice sheets at such low population densities that you can't really detect them with pollen, but they might still be there. Yes, so there are some places where trees might have survived. We can probably say that somewhere in southern places of the ice sheet there might actually been spruce, because we see we see spruce very early, very early appearing, right? Last glacial time we can already see spruce, which should come from somewhere in the south. And Denmark has not been glaciated, and maybe even Doggerland, which is the land that was between England and Denmark. There might have been small spruce stands surviving there. There might have been this stunt trees, not very forest or trees, because then you would have expected to find pollen. But these are like small trees that are small and probably go undetected based on the traditional methods. Then there is this tree in the top in the north, which has always been the question of debate, whether there was trees growing on the west side of the ice sheet near Andoya. Based on our data, we can't really exclude this hypothesis that there were trees there, but what we can say is that if these trees existed, they were not different from the ones that were colonizing from the east. They basically represent these two main clusters, and in terms of population genetics, it doesn't really matter whether they come from the west or from the east, because they are coming from these main clusters. There's nothing special with the clonal trees over there, which is a very interesting result in itself. So if we continue a little bit, the ice sheet decreased in size, right? Because the climate is decreasing. We think that actually the spruce trees tracked the ice sheets, like they migrated with the ice sheet at low population densities towards Scandinavia, and the main spruce development actually happened a little bit later and was very slow. But spruce might have been very fast in the beginning. Yes. So because we find spruce very early in these three different places in southern Sweden, we think that there might have been some movement from spruce into Scandinavia in the south during the late glacial. 14,000 years ago. Yeah. Okay. So yeah, there are some pollen founds of fossil founds and pollen also in the Oslo area around here and in the late glacial, which makes sense if you assume that there might have been some spruce in the south. Which also confirms what Tage Nielsen was suggesting almost 100 years ago, that there was spruce in southern Sweden. Yes, so then at 10,000 years ago, when there was still a little bit of ice in Scandinavia, the date that the clonal trees, the clonal fossils from Kuhlmann are found in Dalarna and Jämtland, is the place where there is now multiple ways of how spruce could have migrated this place very quickly. If you know that spruce was closer to the ice sheet than you think before. Otherwise, the distance that spruce needs to migrate into Scandinavia is a very long time. But if you have very small populations in between that you can't really attack with pollen, you have a lot of spruce potentially close by this site where migration could have happened. There are some other theories as well that there was ice of seeds trapped in the ice sheet that could have kind of survived, and then as soon as there's land available and the seeds start to grow, it's also a possibility. In any case, there's a lot of spruce nearby, not in big populations, not forests, but big enough to actually start to colonize it. So to summarize this whole thing up, like the pollen still also a true signal, right? Because they saw a very clear increase over years, very recently when forest, spruce forest started to develop. So our idea is that it was not really a migration in the sense that you have like spruce forests becoming bigger and bigger, but spruce was already present in most of these spaces, and they were just satellite populations that are kind of started to expand locally when the temperature became hotter and the climate became better for them to develop, which still happened like very recently. Yes.
What are the different colors of the spruces? Maybe I wasn't here when you told about it.
Yeah, so the different colors are based on genetic ancestry. So the blue ones are from a different genetic cluster than the ones from yellow, and they have been separated for a very long time. So we know that these clonal spruce trees in Jämtland and Dalarna are made out of these two ancestral components. Not sure if it makes it that much clearer, but yeah, the colors represent the genetic background. Yeah, so spruce might actually be a pioneering species instead of a late arrival into Scandinavia. And based on our data, we don't really need a Western refuser to actually explain Kuhlmann's results, or like the genetic diversity can always be explained by trees moving in from the east very early. So to just illustrate the point again with the lake from the beginning, when we find pollen from spruce, they might actually coming from the regional signal from the trees that are quite far away from the lake. It's difficult to say that if you find a few pollen grains, whether they come from these background trees or if they come from the trees that are in the catchment. But when we find DNA, we're pretty sure that they are actually trees that come from the catchment. So we can more certainly say whether spruce is present locally rather than regionally. Yeah, so this is my very last slide. So the overall conclusion, like it's very nice, everything, but why are we actually doing this? And it's basically to, if we can determine where taxa actually are much more accurately and see how long taxa are present in climatic conditions, which might not be very optimal. It might be very useful information to put in climate models to be able to better predict where species move or where species can persist in the future, and especially with this current climate change that's going on, this actually might be very valuable data to be added to the whole list of available information based on pollen and macrofossils. So we don't really, with DNA, we can't really replace the traditional way. We can just like complement it and use all the data to make better predictions. Yeah, and okay, and acknowledgement, shouldn't forget this. I'm not doing this work alone, but a whole list of collaborators. I actually don't think I even put everybody in, so if somebody should be there, yeah. And it was most of the stuff was funded by actually by SVS. So yeah, any questions?
You have this kind of rules that on Chico, for example, with resprouting all the time.
Yeah.
And you have that high up in the mountains, and you have it along the east coast in some places.
Okay.
Do you think that's entirely depending on the habitat, or it would be genetic?
No, I think it's entirely habitat. If you look at the genetic diversity of spruce, I should talk a little bit close to the computer. So genetic diversity in spruce is relatively low, and we see based on our results that the trees that are forest trees, basically in the spruce forest, are more or less the same as the ones that are in the that's growing in these stunt forms. So I think all of the form of the morphology is not genetically based, but it's based on the environment that they are in. Maybe there is some local adaptation going on, but we can't really detect this with the methods that we've been using.
Then it should disappear in the mountains with the
Yeah, I actually think they will disappear if it becomes much higher. Then they will form trees, and then when they die, they don't really have this layering anymore. So they will actually disappear. Yes.
So has nobody done some kind of common garden or something to try and induce the clonal growth form? Cold places or something?
I'm actually not sure. I actually think they take a branch from old Tico and plant it somewhere, but I don't remember anymore.
To try and either induce one or the other growth form from seeds or from clonal
I
material.
actually don't know, but you say these things about they took some branch from old people.
Yeah, I think they brought it to some hospital in the south, and then they grow it as a tree.
Or wouldn't they try and grow forest trees in very cold places and see if they become clonal?
I'm not sure if this is done. No, it's a good question. But you have a lot of spruce hatches, so you can form them whatever you like, and then you can create different. You don't have to have them grow into a tree.
Can you repeat the question? So
Yeah, if you read the
Oh, okay.
question on your head.
you don't
Yes.
have to-
Yeah. Any more questions? Yes.
I have one on the more modern times on this. As you showed that you had the third version of spruce down in the Baltic states, Belarus, Poland.
Yeah.
And as this has been the ones that has been planted in Sweden the last 40 years,
Yes.
solidly, I don't think it's nearly any Swedish or Scandinavian spruce that has been planted since the early 80s. So that is. Did you have the markers on that? You should see if you get some infection of that thing, because that pollen will go widely. So perhaps those old ones, of course, they are still what they are, but the forest trees below the tree line, how much that will disturb the pictures. Do you have any idea of that?
Yeah. So I tried to summarize this question into one sentence, I guess. So the question is how much input is coming from the planted trees in the area rather than the ones that are native, right? Okay, so this is actually a good question. Like a lot of trees have been planted, and there has been studies done where you can see where things are coming from Germany, the Baltic states, and it's mainly happening in southern Sweden, I think. So it's not a problem for the clonal trees, right? Because we know they are relatively old, and the trees in the forest that we took, they are very close to these mountain ranges. So I think they are not planted in plantations. I think, and most of the trees we took are very. I assume they are very old. They are much older than the time from when the plantation started to happen in the last 50 years. So I think we don't really have this signal. At least we didn't really find any signal from this. But in the future, you will have a lot of these mixtures going on from all of these different origins that are mixing together. The ones in the plantations, the native stands, they will form some kind of hybrids in the future.
You have been lucky to work at this time, up to 50 or 100 years in the future.
Yeah,
But it shall be much more messy, I think.
I think so, but also we know a lot of the plantations where they have been planted, and based on genetics, we know which individuals probably are mixes between different, because you will have then this extra cluster coming in from the Baltics. Yes. Any other questions? Time for food. Cool. Then thank you very much for your attention.
Thank you.